There are approximately 8 million Ashkenazi Jews alive in the world today. They live in New York, Tel Aviv, London, Buenos Aires, Melbourne, and dozens of other cities across the globe. They include Nobel laureates, Hollywood directors, Supreme Court justices, tech billionaires, musicians, scientists, writers, and roughly half of the entire worldwide Jewish population. They speak different languages. They hold different passports. They look different from each other. Some are religious, some are secular. Some have never set foot inside a synagogue. But roughly 3.5 million of them, 40% of all living Ashkenazi Jews, share something so specific, so genetically precise, that when scientists first discovered it, they had to check their results twice. These 3.5 million people all descend on their mother's side from just 4 women. 4, not 400, not 4,000. 4 individual women whose mitochondrial DNA was passed from mother to daughter, generation after generation, for roughly 1,000 years, and whose genetic signatures are now carried by millions of people scattered across 6 continents. To put that in perspective, that means there are more people alive today who descend from one of these 4 women than there are people living in the entire country of Jamaica, or Croatia, or New Zealand. 4 anonymous women, each of whom probably lived a quiet, unremarkable life in some medieval European village, turned out to be the genetic matriarchs of one of the most influential populations in human history. Nobody knows their names. Nobody knows what they look like. Nobody knows where exactly they lived or what they did with their lives. But their DNA is everywhere. And the story of how 4 anonymous women became the matriarchs of one of the most influential populations in human history is one of the most remarkable revelations in modern genetics. If you find these stories fascinating, make sure you subscribe to Bloodlines and Borders. To understand this story, you need to understand one piece of biology. Every human being has 2 types of DNA. The first is nuclear DNA, the stuff in your chromosomes, which you inherit from both parents and which gets shuffled and recombined every generation. Nuclear DNA is complicated. It is a mosaic of hundreds of ancestors all mixed together, and after a few generations, it becomes nearly impossible to trace any single ancestor's contribution. But there is a second type of DNA that does not work this way. Mitochondrial DNA is contained in tiny structures called mitochondria that float in the cytoplasm of every cell. Here's the critical difference. Mitochondrial DNA is inherited exclusively from your mother. Your father's mitochondrial DNA dies with him. It is never passed on. This means that your mitochondrial DNA is an exact copy of your mother's mitochondrial DNA, which is an exact copy of her mother's, which is an exact copy of hers, going back generation after generation in an unbroken maternal chain that can stretch for thousands of years. Occasionally, a random mutation occurs in the mitochondrial DNA, creating a new variant that distinguishes one maternal line from another. These mutations are rare and they accumulate slowly, which makes mitochondrial DNA an almost perfect tool for tracing maternal ancestry across deep time. By cataloging the mutations, scientists can group people into maternal lineages called haplogroups and trace those lineages back to specific founding individuals. In 2006, a team led by Dr. Doron Behar at the Rambam Medical Center in Haifa, working with Professor Carl Skorecki at the Technion Israel Institute of Technology, studied the mitochondrial DNA of Ashkenazi Jews from 67 countries. They compared their results with the mitochondrial DNA of non-Jewish populations and other Jewish communities worldwide. What they found was stunning. 4 distinct mitochondrial DNA signatures appeared in their Ashkenazi samples with overwhelming frequency. Together, these 4 signatures accounted for approximately 40% of all Ashkenazi mitochondrial DNA. These signatures were virtually absent in non-Jewish European populations, and they appeared only rarely in Sephardi and Mizrahi Jewish communities. The conclusion was inescapable. Roughly 3.5 million Ashkenazi Jews alive today descend through an unbroken chain of mothers from just 4 women. If you find these stories fascinating, make sure you subscribe to Bloodlines and Borders. The next question was obvious. Where did these 4 women come from? And this is where the story gets genuinely controversial, because two major studies came to different conclusions. The 2006 study by Bahar and Skorecki argued that the founding mothers were likely of Near Eastern origin. Their mitochondrial DNA, or mtDNA types, while rare in the general population, showed patterns consistent with migration from the Levant, the ancient homeland of the Jewish people. This interpretation fit neatly with the traditional narrative of Jewish history. Jewish men migrated from Israel to Rome after the destruction of the Second Temple in 70 CE, and from Rome northward into the Rhineland, bringing their wives with them. The founding mothers in this reading were Jewish women from the ancient Near East who traveled with their husbands into Europe and established the communities that would become Ashkenazi Judaism. Then, in 2013, a team led by Professor Martin Richards at the University of Huddersfield published a study in Nature Communications that turned this interpretation on its head. Richards and his colleagues analyzed a much larger data set, over 2,500 complete mitochondrial genomes and 28,000 partial genomes from populations across Europe and the Near East. Their conclusion was dramatic. All four of the major founding lineages, the ones accounting for 40% of Ashkenazi mitochondrial DNA, had their deepest roots not in the Near East but in prehistoric Europe. What Richards was arguing was not that the Jewish people have no connection to the Levant. The Y chromosome evidence tracing paternal lineage consistently shows a Near Eastern origin for Jewish male ancestors. What he was arguing was that the founding mothers of the Ashkenazi community were primarily European women, not Near Eastern ones. In other words, when Jewish men migrated from the Levant into southern Europe in the 1st and 2nd centuries, they married local European women who either converted to Judaism or were absorbed into the nascent Jewish community. These women became the matriarchs of the entire Ashkenazi population. This interpretation remains hotly debated. Bihar and other geneticists have pushed back, arguing that the European haplogroup assignments are based on imprecise methods and that the founding lineages could still trace back to the Near East through intermediate populations. The debate has become one of the most contentious in population genetics, in part because the stakes are not purely scientific. The question of whether Ashkenazi Jews are really from the Levant or really European has political implications that go far beyond the laboratory, touching on questions of indigenous identity, the legitimacy of Zionism, and the relationship between genetics and peoplehood. Scientists on both sides have been accused of letting politics influence their conclusions, and the truth is probably more nuanced than either camp admits. The honest answer is that the science is not settled. Both studies agree on the core finding: 40% of Ashkenazi Jews descend from four founding mothers. Where those mothers came from, Europe or the Levant, is still an open question. What is not an open question is the finding itself. 4 women, 3.5 million descendants. That part of the story is rock solid. But regardless of where they came from, the founding mother phenomenon tells us something extraordinary about the demographics of the Ashkenazi population. The community that would eventually produce millions of people began as something almost unimaginably small. Think about what this means in human terms. At some point, roughly 1,000 years ago, in the early medieval period, the Ashkenazi Jewish community in the Rhineland was a tiny group. How tiny? Geneticists estimate the effective founding population was in the low hundreds, possibly as few as 350 people. From this minuscule community, the Ashkenazi population expanded to roughly 10 million by the eve of World War II, a demographic explosion of staggering proportions. This expansion left a genetic signature that is visible in multiple ways. The extreme frequency of certain genetic diseases in Ashkenazi populations, including Tay-Sachs, Gaucher disease, Canavan disease, and familial dysautonomia, is a direct consequence of this bottleneck. When a population this small expands rapidly, rare genetic variants that would normally be diluted in a larger population become concentrated. A mutation that might appear in 1 out of every 10,000 people in a large, genetically diverse population can appear in 1 out of every 15 or 20 people in a population that grew from a handful of founders. The same founder effect that produced the 4 maternal lineages also produced the elevated disease frequencies. The diseases and the matriarchs are two sides of the same genetic coin. This is also why Ashkenazi Jews show significantly elevated rates of BRCA1 and BRCA2. Mutations, the genes associated with increased risk of breast, ovarian, and prostate cancer. Approximately 1 in 40 Ashkenazi Jews carries a BRCA mutation compared to roughly 1 in 250 in the general population. These mutations are not a sign of some unique biological flaw. They are a mathematical consequence of a small founding population that expanded rapidly while maintaining high rates of in-group marriage. The founders happened to carry these variants, and because the population grew from so few people, the variants spread far more widely than they would have in a larger, more genetically diverse group. The Ashkenazi population also shows remarkably low genetic diversity on both the maternal and paternal sides compared to other populations of similar size. This is exactly what you would expect from a community that maintained high rates of endogamy, marrying within the group for centuries. The four founding mothers did not just contribute their DNA to the population. They contributed a disproportionate share of it because the community was small and because it stayed small and insular for centuries before the great demographic expansion of the late medieval and early modern period. There is something almost biblical about this finding, and the parallel has not been lost on the researchers. In Jewish scripture, the entire nation traces its origin to four matriarchs: Sarah, Rebecca, Rachel, and Leah. These four women, the wives of Abraham, Isaac, and Jacob, are considered the founding mothers of the Jewish people in religious tradition. The discovery that the Ashkenazi population literally traces 40% of its maternal DNA to four founding women is not a confirmation of the biblical narrative. The timelines are completely different. The genetic matriarchs lived roughly 1,000 years ago, not 3,000. But the structural parallel, four women giving rise to a nation, is striking enough that even secular scientists have remarked on it. Professor Skorecki, who supervised the original study, noted the resonance carefully. The genetic founding mothers, he said, came from lineages that originated long before the Jewish people existed as a distinct group. They probably came from a large Near Eastern or European gene pool. But through the accidents of history, migration, conversion, marriage, and survival, their mitochondrial DNA became the dominant maternal signature of an entire people. They did not choose to become matriarchs. History chose them. What does this mean for someone who is Ashkenazi Jewish today? It means that your maternal lineage, the chain of mothers stretching back through your mother, your grandmother, your great-grandmother, and beyond, has a roughly 40% chance of connecting you to one of four specific women who lived approximately 1,000 years ago. You share a direct, unbroken maternal line with millions of other people you have never met, spread across dozens of countries, speaking dozens of languages, living vastly different lives, all connected by a thread of mitochondrial DNA that has been copied faithfully from mother to daughter for 50 generations. You do not know her name. You will never know her name. She left no diary, no grave marker, no record of her existence beyond the DNA she passed to her daughter, who passed it to hers, who passed it to hers, in an unbroken chain that stretches from a medieval village somewhere in Europe to wherever you are reading this right now. She could not have known what she was starting. She could not have imagined that her descendants would number in the millions, that they would win Nobel Prizes and compose symphonies and decode the human genome and survive the worst genocide in human history and rebuild their lives on 6 continents. She was one woman. She lived and she had a daughter, and that was enough. 4 women, 3.5 million descendants, 1,000 years of unbroken maternal inheritance. Pogroms could not break the chain. Expulsions could not break it. The Inquisition could not break it. Even the Holocaust, which murdered 6 million Jews and reduced the Ashkenazi population by 2/3, could not break it. The chain held. Mother to daughter, daughter to granddaughter, through every catastrophe that history could throw at it. And today, in laboratories and clinics and DNA testing kits around the world, the evidence of those 4 women's existence is being read for the first time, 1,000 years after they lived, by the descendants who owe their existence to them. That is not a metaphor. That is what the DNA says. And sometimes the science is more extraordinary than any myth could ever be. If you find these stories fascinating, make sure you subscribe to Bloodlines and Borders. Thank you for watching. If you want to go deeper, the next video on your screen is one you will not want to miss. I will see you there.